இது நம்ம ஐயா கோச்சிங் சென்டர். வாங்க படிக்கலாம்.
இந்த வீடியோ உங்களுக்கு பிடிச்சிருந்தா லைக் பண்ணுங்க, ஷேர் பண்ணுங்க, சப்ஸ்கிரைப் பண்ணுங்க. Thank you.
Hi everybody. Today let us see monohybrid cross. What is monohybrid cross? That is only one alternate form of single character is taken. That is known as monohybrid cross. What is the alternate form of single character? That is tall plant and a dwarf plant. Then parent you see here, this is tall plant, pure tall. That means generation after generation they produce tall plants. Same like that, pure dwarf plants, that is generation after generation they produce dwarf plants. Then from this tall plants, the gamete which is produced carries this capital T, and the gamete which is produced by the dwarf plant carries, that is small t. When these two gametes are crossed, that is these parents, when these two parents crossed, and then in F1 generation, what is produced? No, a tall plant is produced, but you see here, this is heterozygous tall. This is impure. This is not pure because it carries both the dominant as well as recessive. But it expresses what character? No, tallness. So it is heterozygous tall. In F2 generation, these heterozygous tall is selfed. That is selfing. You can see here, this is also heterozygous tall. This is also heterozygous tall. And then they produce two types of gametes. You can see here, one gamete carries capital T, another one carries the small t, that is recessive. When in F2 generation, when these two heterozygous talls were selfed, then you can get this ratio. Before that, you must know what is this. This is checkerboard. This was first used by Reginald C. Punnett. This was first used by Reginald C. Punnett. Year 1875 to 1967. What is this checkerboard? This is nothing but representation of generation to analyze in the form of symbols of square. Usually you can see here, male gamete lies horizontally. Don't forget, male gamete lies horizontally. Female gametes lie vertically. Okay. Now you can see what the generations we got, whether tall or dwarf or heterozygous tall. You can see capital T, you can get this. This is pure tall. This is pure tall. And capital T, small t, this is heterozygous tall. Then again capital T, small t, heterozygous tall, and small t, then homozygous dwarf. So according to phenotypic ratio, you can see here, these three are tall. According to phenotypic expression, these three are tall. So and then one is dwarf. So the phenotypic ratio is 3:1. But when you see the genotypic ratio, see, this is pure tall 1, and this is heterozygous tall, that is 1 plus 1. 1 plus 1, this is 2, and 1 pure dwarf. So according to genotypic ratio, 1:2:1. In F2 generation, we got the phenotypic ratio 3:1, genotypic ratio 1:2:1. Suppose F3 generation, when these are selfed means capital T into capital T, all are tall. Same these when selfed, that is capital T into small t, capital T into small t. Again same ratio you can get 1:2:1. That is 3 tall and 1 dwarf. And when this is selfed, small t, small t, all are dwarf. In F3 generation. Next we will see Mendel's principle of inheritance or laws of inheritance. See, Mendelian principles are the rules of inheritance, and it was first discovered by Mendel. How many principles are there? No, 4 principles or 4 laws of inheritance, based on which cross monohybrid and also polyhybrid crosses. Now we will see the first 3 laws for one gene inheritance. one gene inheritance. One gene inheritance. Okay. See, in monohybrid cross, how many contrasting character he has taken? He has taken only single pair of only single contrasting character he has taken. Each character is controlled by a pair of allele or factors. See, suppose this is tall, means each character is controlled by a pair of allele, and also see this is what dwarf, controlled by a pair of allele. So on the basis of observation of monohybrid cross, Mendel proposed that is a set of generalization or postulates which resulted in the formation of three laws. Let us see one by one. Three laws of inheritance. First one is principle of paired characters. First one is principle of paired characters. Principle of paired characters. Principle of paired characters. See, usually a character is represented genetically in an individual by two unit factors present on the same locus in two homologous chromosomes. See, these are the two unit factor. That is the gene which is located on the same locus in two homologous. This is the homologous chromosome where this is located. This allele is located on the same locus in the two homologous chromosomes. So these can give rise to three types of combination. For example, these can give rise to three types of combination. That is capital T, comma, capital T, small t, and small t, small t. This we have previously seen in monohybrid cross. So this is known as principle of what? Paired characters. That means a character is represented genetically in an individual by unit of two factors present on the same. This is the factor, unit of two factors that is present on the same locus. They are located on the same locus in two homologous chromosomes. Then second one is law of dominance. Second one is law of dominance. Law of dominance. In the monohybrid cross, we have seen that is when pure tall is crossed with the pure dwarf. What is the F1 generation? F1 generation is heterozygous tall. F1 generation is heterozygous tall. Heterozygous tall. But phenotypically, this plant is tall. So which character is hidden and which is not expressed is, even though dwarf gene is present, dwarf character is not expressed, the plant shows a tallness phenotypically. So which is masked here, this dwarf gene is masked. Masked means it is not expressed. So that is known as law of dominance. Even though it carries the recessive gene, that character is not expressed. The plant is phenotypically, it is tall. So this shows that, so which is dominant, tallness is dominant, and that character is alone expressed in F1 generation. The next one is third law, law of segregation, law of segregation, or law of purity of gametes. Law of purity of gametes. What does mean by law of segregation? See, in the F1 generation, the plant is what it shows what phenotype? Dominant phenotype. That is what it is, tall. Then what happened to the recessive phenotype? It is not expressed in F1 generation, but this recessive character is expressed in which generation? No, F2 generation. We can see here. See, when this is selfed, when heterozygous tallness is self. Previously we have seen this in a monohybrid cross. That is, you see here, that is capital T, small t, capital T, small t, and this is male and female. Then see, In F2 generation, which character is which is masked in the F1 generation is expressed in F2 generation, that is what? Dwarf. And the phenotypic ratio is 3:1. Genotypic ratio is 1:2:1. Okay, you can see here, this in F1 generation, that is the dominant gene as well as recessive gene do not blend. Instead of that, they remain together. But during gamete formation, see here, during gamete formation, the gene segregate. This is one gamete, and this is one gamete, and this is one gamete, this is one gamete. So during gamete formation, these factor or these gene, that is, they segregate during the formation of gamete. Then so only this dwarf character, which is masked in the F1 generation, is expressed in the F2 generation. Another thing you have to see, this is this law of segregation. This is segregation, and law of segregation is otherwise called as law of purity of gamete. What is meant by purity of gamete? So this is one gamete. This gamete carry only one allele, never both. Never both. That is wrong. It always carries only one allele. That is, maybe a capital T. This gamete may carry a small t, and same like here also, capital T or a small t. And so only the gamete is always known as pure. So according to this, he proposed that this law, that is law of segregation, law of purity of gametes. Usually, which is Mendel's first law? Means Mendel's first law is law of segregation or law of purity of gametes. Purity of gametes. That's all today's class. Thank you.